Welcome to our podcast on the respiratory system. I'm going to try to squeeze it all into this one podcast. And certainly in the beginning, we're going to have some review of anatomy. But as we get into the histology, we're going to identify the regions histologically that differ in the conducting and the respiratory zones. And there are a number of cells that we're going to have to identify throughout the respiratory system that will help us understand what level we're in. Then we're also going to look at the smaller passageways, the smaller portions of our conducting and then respiratory zones, the bronchioles. And you're going to need to compare and contrast the primary, terminal, and respiratory bronchioles. And then we'll identify the components of the very critical blood-air barrier that allows for gas exchange. So the conducting portion, the anatomy of our respiratory tract, air can either enter through the nasal cavity or the mouth. And then they're going to join this common passageway in the back of what we call the throat, which is the pharynx. And that can be broken up into the nasopharynx, that portion that's more superior, adjacent to the nasal cavity, the oropharynx, which is the back of the mouth, and then the laryngopharynx, which is the junction of the two together in what we then properly call the next portion, or the larynx. Now, the larynx is going to be that component where we find our vocal cords, but it's also going to be that structure where we have the epiglottis, which is going to assist in directing the movement of air or water and food particles either into the trachea or the esophagus. For the respiratory system, the next passageway will be trachea. Then, as we continue with the conducting portion of the passageway, and again, there's no gas exchange here. These are simply moving air from one place to another. So then we go from our trachea to our primary bronchi. This is where the single trachea splits in two. One goes to the right, one goes to the left. Then we're going to branch again into secondary bronchi. That branches again into tertiary bronchi. And these are also referred to as segmental bronchi, and they're going to go to a bronchopulmonary segment. There are 10 on each side, 10 on the right, 10 on the left. Then we're going to branch into bronchioles. And we have a number of order of branches, and we're not going to go through every single one of these. But we have primary bronchioles, secondary bronchioles, and those are all histologically going to be very similar. But we get to terminal bronchioles, and it's not that their histology is going to differ that much from the preceding bronchioles, but that's the last portion. The terminal name we have there is that's the final portion of our conducting system of the airway. Here we have an illustration of the upper portion of the respiratory system. Here we can see the nasal cavity and the oral cavity that connect in the back, or the dorsal part of the throat here, as the pharynx. Again, this is nasopharynx, oropharynx, and this would be the laryngopharynx as we move down. Here we have the larynx proper, which divides food going down into the esophagus via the epiglottis, or air that moves around the epiglottis and slides into the singular trachea. Now, as we get into the pulmonary tissue itself, the intrapulmonary tissue, and down to the levels of our bronchioles and the last portion of the conducting system, the terminal bronchioles, we then get into the respiratory portion. These are the final steps, and these are the steps at which some external respiration can occur. And the first part of that's going to be respiratory bronchioles. Then we move into alveolar ducts. Then this common sort of hallway or passageway created by these small bubbles of alveoli called alveolar sacs, and then the alveoli themselves where we do have external respiration occurring. Here we can see these end portions. We have terminal bronchioles that would happen way up here. And as we move down, we move into respiratory bronchioles. They're respiratory because you can see that they do have some alveoli that are attached to the bronchiole itself. Bronchioles have these spiraling, smooth muscle ribbons that we can see. Then we move into a duct that is just simply lined with alveoli. This is that common hallway. And then here we have a sac, which is composed of just these globular units of alveoli. If we look inside, we can see inside of an alveolar sac, the alveolus. And each one of these is not independent of the other because they have pores that interconnect neighboring alveoli to each other, and that's to assist in the equilibration of the air pressure. So here are the functions of those units we just talked about. We mentioned the conducting portion is just the movement of air. but more than just a way to move air from the outside environment down to the deeper parts of the lungs. We're going to filter the air, humidify, and warm the air as we move in. And that's particularly important on cold days, like hopefully we'll be having sometime soon. The filtering of the air is going to be a process of the mucus that's produced and the ciliated cells that are going to move that debris-laden mucus out of the airway, and then the humidification occurs just because the primary part of the airway is going to be water. Now, as we look at the conducting portion, we're going to start looking at olfactory epithelium, and this is going to be the roof of the nasal cavity, the nasal septum, and this conchae, these folds that are on the lateral portions of our nasal passageways. And so as we look at our olfactory epithelium, the mucosa is pseudostratified ciliated columnar epithelium. Now, this is going to be typical of all of our respiratory epithelium. So just get used to referring to pseudostratified ciliated epithelium as respiratory epithelium. Now, the mucosa is going to be divided into a couple of different cells. We first have our olfactory cells, and these are going to be partly bipolar neurons. And we're going to have this apical cilia, which are the dendrites, and the basal axons that are going to send the stimuli to other axons. And it's actually these basal axons that make up the olfactory nerve proper. But as we look at the dendrites, you're going to have a number of these on the surface. They're going to be bathed in this fluid that's produced by serous glands that we'll look at as we get into the deeper portions. And so the chemicals, the odorants, they're going to dissolve in these watery serous secretions and then bind to odor receptors on these apical cilia to begin this process and transduce that chemical signal into an electrical signal that will be an action potential. So here we kind of looking at things upside down is what we're used to. Here we can see the olfactory epithelium. This is going to be on the surface. These will be our olfactory bipolar neurons. Here we can see the olfactory cilia on the surface. These are going to be bathed in that serous secretion, and these are going to come from those glands, and these are going to be Bowman's glands. We'll talk about those a little bit later. But as the odorants bind to the receptors, that's going to send the signal down through the cell, down to the axon, and here we see the filia olfactoria. This is going to be our olfactory nerve, and this is going to go to the olfactory bulb and then to the somatosensory portion of the brain. Here's an actual light level micrograph of what we're going to see, our olfactory epithelium. Our cilia across the surface. Here's our pseudostratified epithelium, our olfactory cell. We have our basal regenerative cell. Now, we're going to have a number of cells present here. They're going to be difficult to distinguish. We're going to have our ciliated receptor cells, but we're also going to have supportive cells that are called sustentacular. They just sort of group these all together as olfactory cells here. The regenerative or basal cells, they're pretty easy to see as the nuclei are fairly low down here at the surface. Here we see the lamina propria, and we've got some of these serous type glands that are going to be present down here, these Bowman's glands, and we'll see more of those as we look at additional slides. So we've got our olfactory cells, the bipolar neurons. Here are the supportive cells, are sustentacular. They're going to have microvilli on the surface, and they are going to be the ones that provide this physical support. They're going to assist in the nourishment of these cells. They're going to electrically insulate these cells, and actually to help maintain this sort of distinct environment in the olfactory lumen versus down in the lamina propria. These sustentacular cells are going to form zonula occludens with the olfactory cells, and therefore tight junctional complexes, so the material can't seep down between the cells and into the lamina propria. The basal cells, these are just our regenerative cells, and we saw them at the bottom. They say they're rather pyramid-shaped, but you're mostly just going to see the nucleus of these cells. These regenerative cells can reproduce the bipolar neurons and the sustentacular, and their lifespan, less than a year, which is fairly slow. We just got done with digestive tract where we're looking at 5 to 7 day turnaround. Less than a year is still fairly long for cells in the body. Now, the lamina propria, which is beneath our mucosa, this is again where we see our dense irregular connective tissue, which again we just now associate with lamina propria. This is where we're going to have our Bowman's glands, serous glands, tubuloacinar, and this is going to provide this fresh flow of fluid washing over the olfactory cilia And that also prevents overstimulation. When you bind an odorant to the receptor, this continuous flow of these serous secretions are going to help to wash that out. So you're not overly stimulated by any one particular chemical. Now we're also going to see that in the lamina propria, this is where we have a lot of plasma cells possibly that produced IgE. And these IgEs binding to the mast cells, these are going to be the ones that create all the havoc by their secretion of histamines and heparins that cause inflammation, especially in the nasal cavity, which unfortunately I'm experiencing even as I try to do this podcast. Here we have another illustration. I don't like this one as much. It's not quite as clear. We can see the pseudostratified nature of the epithelium. You get a hint of some cilia, but all of this material in the lamina propria, and it's very difficult to even see our flowing collagen fibers. You can in some places, but this is mostly serous acini that you see here. Here's another. Here's some more serous acini. These are all just filled with Bowman's glands that we have present down in the lamina propria. Now, the pharynx, once again, as we move down out of the nasal cavity, we are going to move into the back of the throat, the nasopharynx. Again, we've got our pseudostratified ciliated epithelium. This is where we're going to find our adenoids in the nasopharynx. The oropharynx, again, this is going to be a continuation of our oral epithelium, and that's going to be stratified squamous epithelium. And then when we get to the junction, the laryngopharynx, this is going to be a place where we still have this stratified squamous epithelium, but it's one of the few places where we're going to see that before we move back into our respiratory type epithelium or our pseudostratified ciliated epithelium. The larynx, basically this is where we are going to divert the solids and liquids and prevent them from entering the trachea. And that's going to be a function of the epiglottis. But we also have the function of phonation here, the process of making sounds. So the larynx is supported by cartilages, and we're not going to go through all of the different cartilages we have there. We have the cricoid cartilage, and that's going to be the one that provides the Adam's apple, the enlarged Adam's apple there in males. But we also have the epiglottis at the top, and the epiglottis is going to cover over what some refer to as the laryngeal adytus. And you can also refer to this as the glottis, the opening of the trachea itself. So it's going to cover over the trachea so that it creates almost like a ramp for solids and liquids to flow past this laryngeal adytus and into the esophagus. Now, as we move past that point, now we're going to get to a place where we have these folds on the lateral walls of our larynx. Now, the most superior are going to be referred to as the vestibular folds. Now, these are going to be covered with our typical pseudostratified columnar ciliated cells like we see in the respiratory system. They're going to be supported by very dense connective tissue. They're very firm. They're immovable. These are going to be to protect the underlying vocal cords, which are much more delicate. Now, we know the vocal cords, they are going to be controlled by various muscles that are going to pull them to different degrees of tautness, and that's called the vocalis muscle. And these are also these vocal folds, or vocal cords as we know them. They're going to be reinforced on the edge by this dense, regular, elastic connective tissue. And this is going to allow for the vibration that occurs so that not too much damage. You're not going to rip the edges of the vocal cords. Have you ever seen an old flag that's been waving in the wind, or especially a storm for a long time? The edges get frayed. So this vocal ligament is going to prevent that fraying from happening on these delicate edges of the vocal cords. Now, the way you can tell the vestibular folds from the vocal folds: vestibular folds, pseudostratified ciliated epithelium; vocal folds, stratified squamous epithelium. So here we have, looking down into the larynx, we actually have an illustration. Here we can see illustrated the epiglottis. Here we have a vocal fold, and this is going to be our vestibular fold. They call this ventricular fold in this old illustration. And then here we have the opening going into the trachea, and this is going to be the laryngeal adytus, and this is going to be the glottis, actually, the opening of the trachea. And the vocal ligament would be right along the edge of this material that we see here, right along the edge of the cords itself. Now, Past the larynx, of course, the cricoid cartilage, that's going to be that portion of the larynx that really defines it. But past the cricoid cartilage, that's where we're going to begin the trachea. It's going to be about 12 centimeters long, 2 centimeters in diameter. And we are going to have 10 to 12 U-shaped hyaline cartilage rings, these tracheal rings of cartilage that are going to provide support along the length of the trachea. Now, they say they're U-shaped or C-shaped, and it's going to be between the open arms of the U or the C that we're going to have this trachealis muscle. Now, this is the muscle that's going to be able to contract and narrow the airway or dilate so that you can expand the airway. Now, that's going to create a somewhat flattened surface, and that's going to be on the posterior or the dorsal part of the trachea facing away. What we see on the throat, that's our ventral surface. So in an illustration, as we look at a cutaway through the larynx, we can see this is the epiglottis. Here we're going to have the vestibular folds, and then we've got our vocal cords, which are just beneath. You can see these are superior, these are inferior. And as we move down, we've got the cricoid cartilage, and then here we have our tracheal rings of hyaline cartilage as we move further and further down. Looking at sort of an above view, looking down at our U-shaped or C-shaped tracheal rings. Here you can see our hyaline cartilage. You can see our ciliated epithelium in this illustration. And here we have our trachealis muscle. And you can see when you contract this muscle, it pulls these arms closer together. And it can actually contract the diameter and the area of our trachea by about 20%. Now what I'd like to show you is a YouTube video clip that I found of a laryngoscopy in which an instrument was inserted down past the larynx into the trachea, showing the tracheal rings, and down to the bifurcation of the primary bronchi. Now we want to refine a little bit our understanding of respiratory epithelium. Now we're out of the olfactory epithelium, so we left those bipolar neurons behind. Now we're looking at the mucosa of the respiratory epithelium, especially in our trachea, in our bronchi. And what we're going to see is there are a number of different cell types. Certainly as we look at these, we're going to have goblet cells, ciliated columnar cells, and then our basal cells. These are going to represent the bulk, 90% of what we find present in our respiratory epithelium. As always, the basal cells, regenerative. Goblet cells, we know, are mucus producers. Those ciliated columnar cells are present for movement, to move this mucus that becomes laden with debris, move it up toward the larynx and the epiglottis before it transitions and moves into the esophagus. That's how we keep clean the respiratory system. We also have what are called brush cells, and maybe about 3% of these. These are going to be taller, a little bit taller than normal, with tall microvilli, not cilia. And it suggests that these may be goblet cells that have released their material. They're not extremely sure what all these brush cells are, but maybe 3% or so. 3% or so serous cells that are producing a serous-type enzymatic secretion, and then we do also have some of our DNES, hormone-producing cells in abundance, maybe 3 to 4% to round out all the cells of our respiratory epithelial mucosa. Once again, we can look and see in a light level micrograph, we can see our pseudostratified nature that we have. We can see our ciliated surface. Here we see 2 goblet cells in this particular illustration. Here we can see some of the cells that are present, the very tall microvilli, which are going to be present, possibly those cells that are the goblet cells that have secreted their material, and then the shorter ciliated type that we see present. Now, lamina propria is fairly innocuous, loose fibroelastic connective tissue. Then the submucosa, this is where we're going to continue to have these glands, mixed serous, maybe some seromucous glands that are present there. This is where we're also going to have some lymphoid elements, some of that diffuse tissue, the GALT, or if we're in a bronchi, we can refer to that as BALT. GALT is for the gut. BALT is bronchiolar diffuse lymphatic tissue. Here in this almost artistic illustration, it's a very different type of stain, but we can see some of those seromucous glands, the Bowman's glands. We can see the acini down in the submucosa, and we can see the ducts projecting up and emptying into the lumen. The adventitia, this is where we're going to find our tracheal rings in the trachea. And again, that's hyaline cartilage rings. Then we're going to have the hypodermis, which is going to happen ventrally. And since the trachea is going to be ventral, we've got the esophagus dorsal. Then this adventitia is going to blend with the adventitia of the esophagus dorsal to the trachea. Now, here's where we move down what is referred to as the bronchial tree. As we leave the trachea, we enter into our primary bronchi. Now, this is extrapulmonary. The trachea, the primary bronchi, are the extrapulmonary conductive portions. Then as we enter the lung tissue itself, these are then going to be the secondary bronchi, which are also referred to as lobar, the tertiary bronchi, which we talked about, and we'll touch on these even more. Then we get into the various stages of bronchioles before we get into the terminal and the respiratory bronchioles. And again, remember, terminal means the terminal portion of the conducting system before any external respiration can occur. So primary bronchi, these are going to the right and to the left lobes of the lungs. These are going to branch then into our secondaries, which are the trifurcation on the right side, and then we're going to have the bifurcation on the left. Remember, on the right side, we have 3 lobes, and on the left side, we have 2 because the heart shifted kind of over to the left side, and you can recognize that here. In this illustration, we have our trachea, our 2 primary bronchi, then we have our 3 on the left, and we have our 2 on the right. I'm sorry, 2 on the left, 3 on the right. Now, the secondary bronchi, when we look at these, the primary bronchi histologically are going to look fairly similar to our trachea. When we get to the secondary bronchi, we're still going to have the cartilaginous rings. They're going to completely surround the lumen of the bronchi. We're going to have 2 layers now of smooth muscle, and these are spiraling smooth muscle. We kind of saw that in the illustration early in our slide. And again, we're still going to have our seromucous glands and the diffuse lymphatic elements in the lamina propria and submucosa. So fairly similar to what we see histologically with our trachea and primary bronchi. The only way you could distinguish a primary bronchi from a secondary bronchi is one is outside of the lung tissue and one is within the lung tissue. So here we have an example of lung tissue, and you can see it's mostly space, just mostly empty airspace, all the alveoli that make up this area. We can see our bronchiole, they're always going to be folded up. That lumen's going to be really folded up. And so these smaller areas are going to be bronchioles, but these larger areas that are completely surrounded by hyaline cartilaginous rings that we can see here, and our spiraling smooth muscle, these are going to be our secondary or tertiary bronchi. And it's going to be difficult at this point to tell secondary or tertiary because we were within the lung tissue. What we could say is this is an intrapulmonary bronchus, and that's probably the best we can do at this point. Now, the tertiary, these are going to be 10 per side. I know we had 2 on the left and 3 on the right with secondaries, but now everyone's going to branch into 10 segments, and these are going to go to 10 bronchopulmonary segments. So as you look at the different colorations on the lobes of the lungs themselves, these are going to be completely isolated compartments. So if one bronchopulmonary segment is damaged, it's not going to deflate the entirety of the lung itself. It's not going to damage any of the other areas. So these secondary bronchi, tertiary bronchi, 10 go to the left and 10 go to the right. So once again, in our illustration, we can see our trachea. We can see our primary bronchi. Then we're going to have secondary bronchi. And now we get down to the level of our tertiary bronchi. And they're going to be 10 on each side. Then the smaller ones we get down, these are going to be the bronchioles. So once again, as we're looking at our bronchi, we're going to have some cartilaginous elements associated with our respiratory mucosa. And we're going to have our spiraling smooth muscle on the external surface. So again, we're still looking at bronchi. Now, as we get down to our bronchioles, this is going to be about the 10th through the 15th generation of branching by the time we get to our bronchioles. And what we're going to see is we define bronchioles by having a diameter, a luminal diameter less than a millimeter. Now, the large bronchioles, they are going to have ciliated simple columnar epithelium. Notice we have moved away from our pseudostratified. Now we're simple columnar, but we're still ciliated. Now, we do have a few goblet cells that are present, but as the diameter gets smaller and smaller and smaller, the mucosal layer is going to get more and more simple, fewer and fewer cell types until we get down to the point that we have our our air-blood barrier. So when we get to the small bronchioles, by then we have simple cuboidal, and they may or may not have cilia here, but we're not going to have any goblet cells, and we're going to introduce a new cell type that we'll talk about later. And these are the Clara cells that are going to be present in our small bronchioles. So now as we look at this histological slide, immediately we know we're in lung because we can see mostly this empty airspace. These are alveoli that we have these structures embedded within. Now we can recognize our simple layer of epithelium. So it's difficult to see cuboidal versus columnar at this magnification. Maybe we've got a little columnar happening here. We got quite a bit of smooth muscle, but notice we don't have any cartilaginous elements here. So we know we are looking at a bronchiole of some size. We know it's not a respiratory bronchiole. It's way too big for that, and we don't see any exits for alveoli off that continuous lumen. So at this point, we can know we are looking at a bronchiole. And we know it's not a respiratory. We just don't know at what. We know it's not a respiratory, and it's not likely to be a terminal based on how I'm going to quiz you, and we'll get to that in just a minute. Now, Clara cells, this is a new cell type. When we look at these, they're going to be columnar, but they're going to have very dome-shaped apical surface with short microvilli. So these, in some ways, may have that dome shape like we saw with transitional epithelium, but not nearly that big. Now, Clara cells are going to protect the epithelium by secreting a surfactant-like material. Now, I can't stress that enough. Do not confuse this secretory product with surfactant. This is not surfactant at all. It's a protective lipoprotein secretion, more so than necessarily the surfactant that's going to be used to disrupt the surface tension that's produced by another cell type. All right, so please don't confuse that. Now, the Clara cells in some ways are also going to be able to be regenerative, and they can divide into new cells, not just new Clara cells, but new epithelial cell types as well. So once again, as we look in our micrographs, we can see cells that have these fairly long microvilli, but then we have the cells that have these really short microvilli that we see in here, and these are going to be our Clara type cells. These are probably going to be our dust cells or macrophages that we have present in here. In a light level micrograph, we can see we've got a blood vessel filled with red blood cells here, but then we have these fairly tall columnar domed shape, a lot of these dome shaped cells that are present associated with this fairly thin-walled bronchiole that we have here. Say bronchiole because we don't see any cartilaginous components out here. So these are going to be the Clara cells present on that surface. Now, also associated with our bronchioles, no glands in lamina propria, no cartilage, some spiraling smooth muscle that will become fewer and fewer in number as our luminal diameter gets smaller and smaller. Once again, the message here is the wall of the passageways are getting thinner and thinner and more simple as we get closer and closer to the position where we're going to have external respiration. Now we finally get to the terminal portion of our conducting system. These are the terminal bronchioles, and these diameters are going to be less than 0.5 millimeter. Now the epithelium is going to be cuboidal, and we may have a few clara cells, but really other than that, all we're going to have are 1 to 2 layers of smooth muscle present in the lamina propria. That's really going to define our terminal bronchioles. So you can see we're getting very close to the end of our airway, and if this is the last portion of our conducting system. Then we're going to get into the respiratory bronchioles and our alveoli complex that we have. So once again, histologically, when we look at this particular micrograph, we are first struck by the whiteness, all the white empty spaces, and that's at first blush going to let us know we're looking at lung tissue, and all these empty spaces are alveoli. Now, we're going to start looking at this. We definitely have a bronchiole. You can see our epithelium. You can see our smooth muscle. We have no cartilage components, and you see how it's continuous. There are no breaks in the wall of this bronchiole. But just looking at this portion alone wouldn't tell us that we're in a terminal bronchiole. The fact that we're leaving this and we can follow it continuously until we get to this point or this point, we still have smooth muscle, but there, do you see that break? And that break, possibly a break there. Over here, we've got a break. There, we've got alveoli coming off. This is going to be the beginning of the respiratory portion. This is a respiratory bronchiole. You're still going to have smooth muscle, but the occasional alveoli branching off. And as we move from an intact wall bronchiole into a respiratory, that means that this that immediately preceded the respiratory bronchiole, this has to be the terminal bronchiole. That's how you make that. It's more of an associative thing than necessarily a histological reckoning just on the tissue of the layers itself. So the respiratory bronchiole, similar to what we saw with the terminals, however, you have those alveoli that are intermittently interrupting the wall. And that's the first place where we can have our external respiration. So remember, external refers to the exchange of material between the blood and the air that's been brought in from the external environment into these deeper portions of the lungs. That's why we call it external. Internal respiration, remember, is down at the level of the tissues. So here we can see an alveolar sac complex and all the alveoli together. Here's just an alveolar duct. And they're all going to be completely invested with capillaries very closely. So our blood-air barrier is just the lining of the alveolus and the endothelial cell of this very thin capillary that's going to allow the diffusion of oxygen into the blood and the diffusion of carbon dioxide out of the blood and into the spaces of the alveolus to be exhaled. Here you can see a little more detail about that transport of carbon dioxide. Oxygen's fairly easy. It's just on and off a hemoglobin. So here's another look. We get down to our alveolar spaces. Here we have an alveolar duct, which is just a little passageway defined by only alveoli present. And then as we see this smooth muscle layers intermittently interrupted by alveoli, that's how we can know we're looking at a respiratory bronchiole in this area. Now as we get to the end of our passageway, we began our external respiration in the respiratory bronchioles. Now we move into what are referred to as alveolar ducts. Now these only are structures because they are a common passageway to get to the alveoli. So if you stack all these alveoli together and create a tunnel within the middle of them, that's an alveolar duct. And really the only way histologically we distinguish these from alveoli themselves is that surrounding each little passageway to get to alveoli, there's a single smooth muscle, and this is going to help control the passage of air from the alveolar duct into the alveolus itself. And it creates these little smooth muscle knobs that you see present on the passageway or the hallway that's the alveolar duct. Now the alveolar sac, instead of being a long collection of alveoli, it is a round ball or grape-like cluster of these alveoli, and the center of this alveolar sac is referred to as the atrium. So that's the only difference. An alveolar duct is long, and alveolar sac is more rounded like grapes. Here we can see the alveolar duct running right through the center of our illustration. and we have alveoli present off and completely surrounding this common elongated passageway. And then here are single smooth muscular knobs that are present on these controlling areas. Now, in cross-section like this, you just see the knobs, but understand this is a circular entrance. And so we're going to have this single layer of smooth muscle that will be able to expand and contract this passageway. And in cross-section, we'll just see these as two knobs of smooth muscle tissue. Here we can see an illustration. Each one of these little circles in our scanning electron micrograph, these are going to be an alveoli. This would be an alveolar sac, and this is going to be an alveolar duct as we're moving down through here. In this TEM section, we're simply looking at a capillary here. You can see our red blood cells present. We've got a capillary, no red blood cells that we can see here. And this is going to be one of our alveoli. And then we have our lining cells here that are going to help with the gas exchange of material. Now the alveolus, this marks the end of our journey. This is where we have external respiration, and this is going to be the area where we have our respiratory membrane, where those gases can cross from the bloodstream into the air and back again. Now with all the alveoli, approximately 300 million alveoli in the surface area per alveoli, we're going to have over 140 square meters of surface area that we have present within our lungs themselves. Now these alveoli, they're going to have a diameter approximately 200 microns in the terminal portion of the duct and the sac, and these are extremely small structures. Small enough that water surface tension alone would collapse them if we didn't have assistance from this substance called surfactant that we'll talk about in just a bit. Now here we have an older diagram, and I like to include these things from long ago because they didn't have a lot of the technology we have, but they really got things right. Here you can see our respiratory bronchiole that's interrupted here with our alveoli. Then we got our alveolar duct, and they show the smooth muscle knobs on these ends. Then we get into our alveoli, so this is going to be an alveolar sac. But notice also they have so much detail that they show these alveolar ducts, these passageways between adjacent alveoli, that the pressure is equilibrated from one side of an alveolus to the other. Now, when we have the alveoli adjacent to each other, they're going to share a common wall, and we call this an interalveolar septum, and this is where you find those pores, or the pore of Kahn. So there's going to be a lot of capillaries in these walls. That way gases can be exchanged from one side to the other. It's not going to be very thick, but it's going to be rich in those resistive fibers of type 3 collagen that resist the real stress forces that are going to be happening as the lungs expand and contract. Now, that pore of Kahn, this is going to be formed by lining cells from adjacent alveoli coming in contact and creating this barrier. And those lining cells are called type 1 pneumocytes. So type 1 pneumocytes, these are the cells that are going to comprise the respiratory membrane on the air side of our air-gas barrier. These are going to be simple squamous epithelial cells, and they're going to comprise 95% of the surface area of our alveoli. Now, they will have zonula occludens situated between them, and so these tight junctions are going to resemble continuous capillaries that we're going to see on the opposite side of our air-gas barrier in our capillaries. Here we have a light level view, and we're looking at the air in an alveolus, and this lining cell that we see, our typical simple squamous epithelium, that's going to be our type 1 pneumocyte. And then we can see the red blood cells in these capillaries, and it's going to be, these capillary networks are so extremely dense in these interalveolar septa. Here we have another alveoli here, and in this wall we can see all these capillaries that are exchanging the gases back and forth. Now the other major cell type we have are going to be the type 2 pneumocytes. These are also called great alveolar cells, septal cells, type 2 alveolar cells, and they're going to count for about 5% of the total surface area, but they're going to be very abundant. They're just not flat and spread out. They're more rounded and fairly large than we see, and in fact there are more of them than we have in type 1 cells. Now when we look at these cells, we are going to be able to see them, especially at the EM level, we're going to see these vesicles inside that look layered. They're called lamellar bodies, and these are the vesicles that are filled with surfactant. So they produce and secrete the surfactant, but they're also going to be phagocytic in nature in the fact that they can remove excess surfactant from the alveolar spaces. Now, these cells are fairly diverse in their function because they are also regenerative, and they can produce themselves more type 2s, but in some cases they can produce more type 1 pneumocytes as well. So here we see an illustration of our type 2 pneumocyte, especially the lamellar bodies that contain the surfactant. Here we can see the amino acids and the choline that are coming in to make this surfactant-rich material, these vesicles that coalesce together into these large lamellar bodies. Those then fuse with the surface and release the surfactant. They're showing that coating the surface of this cell, but there's going to be enough that's going to coat all the surfaces of all the alveolar cells, type 1 pneumocytes, all throughout this end-terminal portion of our respiratory system. Here's an electron micrograph showing our capillary. Here is a platelet present in our circulatory system, and here we have a type 2 pneumocyte, and here you can see those extensive lamellar bodies that we have present in our type 2 pneumocyte. At the light level, they're very different than what we see with our simple squamous type 1. Here we can see our very large cell, and you can see the vesicles in here. You can't recognize the layering, but these fairly light staining vesicles, those are going to be the lamellar bodies. Now, the other cell type we have, these are our resident macrophages. We call them dust cells. They remove debris, they remove excess surfactant, any material that gets down into the alveolar spaces. These dust cells are going to absolutely gobble them up. And here in this combined photograph of a transmission electron micrograph, we can see our red blood cells and capillaries. We can see type 2 pneumocytes. We can see type 1, the thin membrane that lines and defines the boundary of our alveoli. That's our type 1 pneumocyte. There's a nucleus of a type 1 pneumocyte. And then here in the alveolar space themselves, here we have 3 of our dust cells, or our resident macrophages. At the light level, we can recognize them. You might confuse them a little bit with our type 2 pneumocytes. Here's a type 2 pneumocyte, and you can see all the lamellar bodies. This is going to usually be somewhat free-floating out in the alveolus itself. Those are fairly easy to identify that way. So our blood gas or blood-air barrier, this is essentially going to consist of the surfactant that's being produced by our type 2 cells, the cytoplasm, the thin squamous cytoplasm of a type 1 pneumocyte, the cytoplasm of our very thin continuous capillary endothelial cells, and the minimal basal lamina that we have situated between the two. That's the extent of our blood gas barrier. Here we can see the illustration. This is showing a little fluid building up in the alveoli, but this shows our type 1 pneumocyte, type 2 pneumocyte with its lamellar bodies, and then the capillary showing endothelial cell, basal lamina, the epithelium of our type 1, and then the surfactant. That's our blood gas barrier. It's actually much thinner than that. And if we look at a transmission electron micrograph, here's an alveolus. Here we have the basement membrane between the type 1 pneumocyte cytoplasm and the capillary endothelium. That is our blood gas barrier. Very thin. And another view shows the thinness. Here we have a half a micron. That's the bar spacing you see here. So less than half a micron across, especially in a place like this. Here we have our red blood cell, capillary cytoplasm of our endothelium, cytoplasm of our type 1 pneumocyte basement membrane. That distance is our blood gas barrier.